project is a comparative computational project on Rosmarinus officinales, Curcuma longa, Syzygium aromaticum, Zingiber officinale, and Withania somnifera, studying the effect of plant-based metabolites on the binding affinity between the ACE2 receptor and the perfusion spike glycoprotein. Severe acute respiratory syndrome, or SARS-CoV-2, is a respiratory virus that has directly affected over 23 million people worldwide thus far, dating back to its emergence in December of 2019. There is research regarding traditional herbs being used to ease the symptoms of the virus, mostly targeting inflammation in the lungs, but not much research has been done in regard to how these herbs could potentially reduce one's risk of actually contracting the virus. The virus enters someone's body and causes them to contract it by attaching to the active site of the ACE2 receptor. ACE2 stands for angiotensin converting enzyme 2. The normal purpose of ACE2 is to convert an inflammation inducer, angiotensin 2, into an anti-inflammatory agent, angiotensin 1,7. Either by denaturing or blocking the active site of ACE2, certain substances could potentially be able to lessen the chances of someone actually catching COVID.
With this, our research question is, which protein, the perfusion spike glycoprotein or the ACE2 receptor, will bind more strongly to the plant-based metabolites that we have chosen to test? Furthermore, which of these plant-based metabolites exhibit the best inhibition and neutralization qualities between this glycoprotein and the receptor? The purpose of this study is to contribute to existing data within plant-based medicinal research towards controlling the spread of SARS-CoV-2 by finding an efficient, affordable, and safe way to reduce one's chance of contracting the virus. And our hypothesis is, if the binding affinity between ACE2 and the metabolites and the perfusion spike glycoprotein and the metabolites are calculated using Schrodinger, then the docking value will be higher on average between the perfusion spike glycoprotein and the metabolites because this protein is larger than ACE2 and therefore has a larger active site in which the metabolites can bind to.
In an environment set up by Schrodinger, the computational software we are using, we tested various proteins with metabolites. Each were chosen from a database, hence it being a controlled aspect. The proteins, perfusion spike glycoprotein and ACE2 receptor, were tested with plant-based metabolites from turmeric, cloves, ginger, ashwagandha, and rosemary. The docking score of these was the output of the bindings between ACE2 receptor with the plant-based metabolite and then the plant-based metabolite with the prefusion spike glycoprotein. These were the controls of this experiment were the positive control, which is a docking score of -10, which means it bound fairly well, while a negative control would be a docking score of 10, which means it almost repels the thing it should be binding to.
The procedure goes as follows. First, we must obtain the proteins from the protein database for all the metabolites and import them into Maestro, which is a suite in Schrodinger. We need to perform lig prep on all metabolites and protein preparation on the ACE2 receptor and the spike glycoprotein to change these from 2D structures into 3D structures so they're more easily docked. Then we must generate the receptor grid between each metabolite and each protein to prepare the docking sites. And then we must perform ligand docking between each metabolite and protein pair, and use a tool called XP Visualizer to obtain the docking scores that ligand docking produced. We will then perform basic descriptive statistics followed by t-testing on docking scores to enhance understanding of the relationship between docking with the ACE2 receptor and with the perfusion spike glycoprotein. Our results are shown here in this graph. Our null hypothesis is that there is no significant statistical difference between the docking scores of the metabolites, and our alternative hypothesis is that there is a statistical difference between the docking scores of the metabolites. As you can see here, the statistical test that we used to analyze our data was a t-test, and we used this because our study had two main groups of data that were compared to one another. There is a significant difference between the population mean of metabolites binding with the ACE2 receptor and the perfusion spike glycoprotein. And the p-value is 0.001, less than 0.001. It's important to note that these docking scores are not strong enough to induce these agents have direct effects on SARS-CoV-2, so more research is necessary. It's also important to note that the metabolite dihydrocurcumin and somanon do not have a p-value of less than 0.001, which also indicates that further research must be conducted.
As for some limitations of our experiment, our project is solely computational, meaning that the project does not take into account bodily functions and processes outside of the proteins being used, as well as the state of health of the individual. Some examples of components that cannot be included are diseases, allergies, and symptoms that an individual may have. Our computational project also makes it so that the experiment does not consider environmental factors that could affect an individual's likeliness of catching the virus, such as the prominence and amount of COVID exposure around an individual.
The potential consequences of other molecules and proteins in the body are also not included in the computational software of Schrödinger, which is a limitation. Future aims is that we hope to test more metabolites, test the metabolites with the glycoprotein, and then that with the ACE2 receptor, and then determine if they neutralize or have a stronger inhibition, and find, of course, other herbs that could possibly work. In addition, we hope to test with other receptors in the immune or digestive system, and to test for basically stronger bonds and more like related results, since our last ones were not strong enough.
The implications of this project is to provide a preventative treatment that is readily available and easily accessible to the vast majority of our community. And it supplies citizens of third world countries with affordable preventative options until full access to vaccines is present in those areas, and controls the spread and reception of the virus until full release of vaccines to all citizens is available and possible.